bundle sheet extension, what is meaning of the spongy cell. So quickly we are taking the simple diagrammatic representation for the dicot leaf. We are not taking for the monocot because इतना ज़्यादा कुछ नहीं है in case of monocot. So we are talking about only diagrammatic representation for the dicot leaf anatomy. In case of the dicot leaf, outermost layer, basically these are called as the layer of cuticle. So these are the layer of cuticle, outermost layer. You know very well, cuticle, these are the present toward the lower side as well as the upper side. So cuticle, these are the some waxy covering layer who cover the surface area of the leaf. So cuticle present upper side as well as the lower side of the leaf.
Ma'am,
Huh?
should we write this in differentiation?
No. I'm taking only diagrammatic representation. That's it. No differentiation part because we already covered those part, right? Hmm. Yeah. So exactly below the cuticle, now there is a presence of the upper epidermis. So suppose these are the single cell, compactly arranged. These are called as the epidermis, and exactly below the site, again there is a presence of epidermis. So this one basically upper epidermis, and the lower side presence of lower epidermis. I hope you remember last class we already discussed one specific name for the upper epidermis and the lower epidermis, means upper side and the lower side. I hope you remember that is simply called as the for the lower side abaxial, for the upper side adaxial. So upper and the lower epidermis. Now above the epidermis, now we are talking about toward the upper side. हमने पहले ही discuss किया था upper side of the leaf. Basically these are the dark green color because in this upper side palisade cells are present, and that palisade cell they are having a high number of chloroplast. So we are talking about the specific type of the cell, palisade cells, like this, tightly arranged. Basically they are column shape. So these are the typical structure of cells, that is palisade cells, which is a tightly arranged, compactly arranged. Palisade cells. So I'm quickly giving the name. This white color, nothing but we are talking about toward the upper side, tightly arranged cell, that is palisade cell. And palisade cell, these are the one type of the mesophyll cells. Now toward the lower side, now there is a presence of the spongy cells. Spongy means what? Something a loose kind of the association is present. So suppose toward the lower side, now there is a some loosely packed cells are present, loosely arranged. There is a no any tight arrangement, some intercellular gap also present. So whatever we are talking about the this cell, this one, simply it is called as the spongy cells. And spongy cells, intercellular space are present, loosely arranged, and again the number of the chloroplasts they present very low amount. Now we are talking about the vascular bundle. In case of the dicot as well as the monocot leaf, close vascular bundle is present. Close means what? Cambium totally absent, right? So quickly we draw simple diagrammatic structure for vascular bundle. So suppose you present a vascular bundle, right? Now, एक second. These are the presence of the vascular bundle. Around the vascular bundle, there is a presence of some bundle sheath cells, like this. So toward the surface area, there is a presence of some cells, which is represented by this red color. And such cells, simply it is called as the bundle sheath cells. Bundle sheath cells. Inside the bundle sheath cells, means there is a ring form structure is present, that is a single vascular bundle, and inside that vascular bundle, toward the lower side, there is a presence of some cells. Those are called as the primary phloem. These are the part of the primary phloem. Again, toward the upper side, there is a presence of some another different cells. These are called as the primary xylem. So such cell simply it is called as the primary xylem. And we already discussed this part. In case of the leaf, cambium is absent, so there is a no any differentiation. There is a no any cambium present between xylem and the phloem. So upper side cuticle, lower side cuticle, upper epidermis, lower epidermis. Now at the middle, mesophyll cells are present. Two types of the mesophyll cells are present. Number one, that is palisade cells. Those are the tightly arranged, and number of the chloroplast high. That's why they are located toward the upper side. So that's why leaf upper surface it is a quite dark in color. Lower side, now presence of the another type of the mesophyll cell, that is spongy cells. Spongy cells, these are the oval shape or the spherical shape, loosely arranged. Number of the chloroplast low. That's why when we observe the lower side of the leaf, basically these are the fainting color. Okay, light green color. So leaf, dicot leaf, they are having two different type of the color. Upper side get vary, lower side get vary due to presence of the mesophyll cells, because in case of the dicot, two different types of the mesophyll cells are present. Upper side palisade, dark green, lower side spongy cells, loosely arranged, faint light green. Okay, done. Done. Okay, Yash. Okay, chalo. One point is left, that is what is meaning of bundle sheath extension. Now can you see here? These are the area. uh, hmm. These are the area where red color bundle sheets are present. Red color. Normally what happen? Bundle sheet, normally they present in a single layer. So can you see here? This bundle sheet, check the last point of what we said. Check the last point of the differentiation. Bundle sheet extension, parenchymus in nature, and they generally single layered in case of the dicot leaf. When you compare this with monocot, maybe single layer or maybe more than one layers are present. So now what happened here, we draw only single layer, because in case of the dicot generally single layer bundle sheets are present. But sometime what happen, sometime in case of the monocot, now that bundle sheet cells they are showing extension like this. They are showing extension. So whatever we draw this part, these are called as the bundle sheet extension toward the upper side as well as the lower side. Bundle sheet extension. Similarly, bundle sheet extension. Okay, bundle sheet extension means what? Normally single layer is present, but sometime in case of the monocot, from single layer they can form a multi-layer, two layer, three layer, four layer, depend upon the condition. So bundle sheet extension, these are the mostly present in case of the monocot. Got it? Because in case of the monocot, there is no palisade cells, there is no spongy cells. Commonly, mesophyll cells are present. So it's very common scenario. They are showing extension. So if you want, you can write down here. Such kind of this bundle sheet extension, it is present in case of the monocot leaf. Because in within one diagram, we cover all the part. Because leaf से question नहीं बनेगा. Don't worry, but it's important to your understanding purpose. What is exactly palisade cell and what is meaning of the spongy cells? That's why. Chalo. Yeah. So any doubt from the diagrammatic representation? Any doubt? Palisade cells, spongy cells, both these are the types of the mesophyll cells. Mesophyll cell, these are the living cell rich with the chlorophyll, right? And major function photosynthesis, as well as they are responsible for storage of the food. Now, in case of the leaf, closed vascular bundle is present, that is without cambium, and in case of this closed vascular bundle, they are covered with, they are protected with bundle sheath cells. Sometimes bundle sheet they form a single layer which is present in case of the dicot leaf, while in case of the monocot, that single layer they are showing extension toward the upper side as well as toward the lower side. Sometimes, not always, sometimes they are showing extension. So such condition it is specifically present in case of monocot leaf. Done. Divyanka, Advik, Anam, Aditya. Sam, Shruti. Okay. Yeah, so attendance done. Chalo. Next part, we are talking about the Bulliform cells. If you have module, you can just open and share me that question which is related to Bulliform. Previous year question में होगा देखो. I think 2019 or 18. Just check once. 2019 or 18 question. So we are talking about next cell, that is presence of the Bulliform cells or the motor cells. Next part. Leaf anatomy part is covered. Now we are talking about the second important point, which is a very important, bulliform cells, and that bulliform cells also called as the motor cell. Hmm? Now, in case of this such type of this bulliform cell, these are mostly present in monocot leaf. They are part of the monocot leaf. Example, these are the part of the monocot leaf. Okay, and under this monocot leaf, you can again, you can, if you want, you can write down the example. They are present in case of the poa, as well as they present in case of the maize, because poa and maize both they are belonging to grass family. And grass, what happened, Vaibhavi? Vaibhavi, just try to re-log in, huh? Just once check. Hmm? Yeah. So we are talking about poa as well as the maize. These are the belonging to grass family, because grasses also they are belonging to monocot. Hmm? Another one example, yeah, another one example we are talking about, that is agropyron. These are the another one example which is belonging to monocot. So these are the few example where specifically in case of the monocot leaf, bulliform cells or the motor cells are present. Now, what is meaning of the bulliform cells? First of all, we try to understand, and then we talk about what is the exactly function. Tab tak, please, anyone you have anatomy module, so please open it. Behind previous year question, वहाँ पे question होगा देखो 2018 or 2019 there is a one question related to bulliform cells in grasses. So just check it and share in chat box quickly. चलो, so तब तक हम देखते हैं some more information about the bulliform cells. Basically, bulliform cells, these are the modified epidermal cell. It's modified epidermal cell. You know very well, whenever we are talking about the epidermis or epidermal cell, normally they are living, normally they are a single layer, but now we are talking about very important, normally they are also, sometimes they are also, means if chloroplast is present, that time they green also. But why we are talking about these are the modification of the epidermal cell? Because such type of the bulliform cells, or where the bulliform cells are present, such type of the monocot leaf, basically they having three important phenomena. C stand for, basically such type of the cells they are colorless. Colorless means we are talking about these are the non-green cells. Bulliform cells, basically they are non-green cells. Another one, basically these are the empty cells. And another one, as compared to epidermis, they are quite large in size. So we are talking about CEL as a code word. Bulliform cells or the motor cell, these are the one kind of the modified epidermis cell. Modified, colorless cell, as well as these are the empty, and as well as they having the large in size. Another point, okay Sam. Hmm. Next point we are talking about, when we are talking about such type of the cell, that is bulliform cells. So such type of this, yeah, thank you Hiram. Such type of this bulliform cell basically present on upper side. They are present on upper side of leaf, and you know very well the upper side of the leaf basically is called as the adaxial. So bulliform cells basically they present toward the upper side. Please underline the word upper side. And the next one, as well as they present near to the veins, upper side and near to the veins. These are the condition. So ये हो गया position. We already discussed some example. Now I'm talking about the exactly function, and you have to tell me. I'll tell you the exactly question. So first of all, explain the concept, and you have to tell me the answer without cheating. Mm, okay. Question number 28 है ये. 2019. Yeah, 2019 NEET question. Yeah. Next part, we are talking about exactly function. So what is the function of the bulliform cell? Normally what happen? Uh, when we are talking about the monocot leaf, basically they are very delicate. Just imagine the grasses. Grasses कैसे होते हैं? Leaf, it is very delicate. It is having long, elongated because cambium is absent. Secondary growth also absent. So it's very delicate. So now what happened? Such kind of, अरे हेराम, let me complete ना, पहले ही आंसर बता दिया. चलो चलो, हेराम, explain me what is meaning of bulliform cell. चलो, tell me function. You send me answer, now you tell me the function. हेराम, हेराम, चल जल्दी से बता, what is the function of bulliform cell? बहुत शाना बनता है कभी कभी. Yeah, tell me. चलो. So next part we are talking about what is the function of the bulliform cells. Bulliform cells basically they are responsible for rolling and the unrolling of the leaf depending upon condition. So we are talking about the first condition, if water present in high amount inside the soil, if water present in high amount, that time these are the first conditions. You know very well, if water present in high amount, it means that absorption of the water absorption also they are more, right? If water absorption more, it means that whatever the leaves are there, leaf, that leaf it is get open up, and that open up condition, simply we are talking about the stage that is turgid stage. Turgid means what? It is get swell. So normally if you draw, if water present in high amount, that time the turgid leaf it is become turgid. Turgid means what? Plenty amount of water is present, so whatever the leaf lamina is there, entire leaf lamina they are showing straight structure like this. They're exactly straight, no curling, no bending, exactly straight. Leaf look very fresh because now what happened, water high hai, that's why leaf it is get open up because they maintain the turgid state, and now such condition these are called as the unrolling. Such condition simply it is called as the unrolling. So if water present in high amount, plant absorb more water, they maintain turgidity, they maintain their proper shape, they become swell. मतलब burst नहीं करना है. Swell मतलब what? They having turgidity. We'll discuss that part later. Turgid, flaccid condition. So they maintain their turgidity, and now leaf it is not showing rolling. So normally such condition is called as the unrolling condition. Second condition, if water present in less amount inside the soil, if water present in less amount, that time what happen? This condition basically it is called as the water stress, unfavorable condition, right? And during unfavorable condition, during the water stress condition, during water stress condition, whatever the leaf is present, now that leaf is become flaccid. It is become flaccid. Flaccid means what? Now that leaf it is get rolled. Leaf showing the rolling. Leaf surface, ultimately surface area of that leaf it is get decreased because to avoid the water loss. So such condition, it's an unfavorable condition. So now such condition simply we can say these are called as rolling condition. Unrolling and the rolling. So bulliform cell it is play very important role during on the basis of their water. If water present in good amount, present in high concentration, that time it is a favorable condition. Now they maintain the unrolling phenomena, means entire structure it is exactly straight. But once water stress condition, unfavorable condition, so now slowly whatever the leaf is there, that leaf they are showing curling process like this, they are showing rolling process. Okay, and such condition simply it is called as the rolling condition. So why such condition is possible? Because of presence of the motor cell, because of the presence of the bulliform cell who control rolling as well as the unrolling of the leaf depending upon water. So if favorable condition, unrolling, if unfavorable condition, rolling. Okay, yeah. So any doubt from the bulliform cell? Any doubt from the
Excuse,
bulliform cell?
excuse me, ma'am.
हाँ बाबा.
Ma'am, can you explain me the first function again? I got disconnected that time.
Listen. First point, first condition, we are talking about the function. What is the major function of the bulliform cell, also called as the motor cell? Closing and opening of the leaf, or rolling as well as unrolling of the leaf. If water present in high amount, means favorable condition, water absorb more water, they maintain their turgid state. Turgid means what? Due to water, now they are having proper shape, that leaf it is look fresh, turgid. So when turgid condition, so now entire leaf they are exactly straight, they are showing very fresh in color, fresh in state. So such condition is called as the unrolling. When water is favorable, water is present means favorable condition. Exactly reverse, when water get less, unfavorable condition, stress condition. During the stress condition, now that leaf they undergo the flaccid. Flaccid means what? Now they become shrink, that freshness they get lack because water less presented. So that time what happen? Ultimately that leaf tip where whenever the bulliform cells are present, that bulliform cell they sense the water concentration, and ultimately they can decrease their surface area, they start rolling to avoid water loss. And such condition these are called as the rolling condition. Got it? Rolling and the unrolling. Simple related to save the water. If water present in favorable plenty amount, that time unrolling. If water stress condition, immediately they can decrease their surface area and they try to save water. Okay? Heram, now tell me what will be the answer of question number. कहाँ गया? एक second. Yeah. Question number 20. 28. Yeah, 28. So grass leaves, grass basically they are belonging to which category? That is monocot category. So it is the grass leaves curl inward, means rolling process actually, curl inward during the very dry weather. Select most appropriate reason for the following. So answer will be, answer will be because Flaccidity of the Bulliform cell. Flaccid means what? Cell becomes shrink due to water loss. That time we can use the flaccid state. So due to flaccid
And
of
please
the
scroll up.
Bulliform cell, हाँ एक second हाँ. Due to flaccid condition of that leaf, now such kind of this
Yes,
rolling
and done.
process or curling process can be occurred. Hmm. Chalo, doubt clear? Any doubt? Any doubt? Okay, done. Should I continue last part of this chapter, secondary growth? Quite interesting, very important, and quite confusing also. Chalo, should I continue? Should I continue? Hello. Okay, Rishikesh. Huh. So next part we are talking about secondary growth. Secondary growth. So this part is important because I hope you remember हमने पहले discuss किया था lateral meristem, lateral, which is a specifically present in case of the dicot as well as the monocot. So now in this point under the secondary growth we'll discuss how that wood formation get initiated, again how the secondary xylem, secondary phloem they get formed, what is meaning of cambium, what is meaning of the intrafascicular cambium, interfascicular cambium, everything we'll try to understand here. Okay, so now we are talking about secondary growth, and secondary growth example is very much common in case of the dicot as well as it is very much common in case of the gymnosperm. Please remember in case of the monocot secondary growth totally absent. चलो, so first of all we are talking about what is meaning of this secondary growth. नाम से पता चलता है secondary. Secondary means what? Formation of something secondary tissue. हमने प्राइमरी टिश्यू देखे थे, लाइक मेरिस्टेम, these are the primary tissue, then the parenchyma, collenchyma, sclerenchyma. Now we are talking about some information about the secondary tissue who form at the later stage of the life. They form due to lateral meristematic activity. So secondary meristem nothing but simple definition, formation of secondary tissue when you are writing the paper, don't use secondary like this. Okay, you have to write down sentence secondary पूरा spelling के साथ लिखना है. Don't use this one, it's for class, only for class. So secondary tissue, formation of the secondary tissue due to lateral meristem activity, due to lateral meristem activity. I hope you remember during the first or second class we already discussed presence of the three types of the meristem, that is apical meristem. Under that apical we discuss sam and ram. Again we discuss intercalary meristem, which is a part of the sugarcane, which is a part of the mostly in case of maize, even grasses also. And another one we'll discuss about the lateral meristem, which is a form at the later stage of the life. So यहाँ पे हम बात कर रहे हैं the secondary growth. These are the one kind of the growth due to activity of the lateral meristem, and again it is a part of the secondary tissue. Now what happened when you talk about the secondary growth? So when you talk about the secondary growth, in case of the secondary growth, ultimately the girth or diameter of the plant, girth as well as the diameter of the plant, they always increase. So we are not talking about length increase, we are not talking about any size will be increased, we are not talking about the elongation, we are talking about the girth or diameter of that plant, they get increased. And when you consider diameter or the girth of the plant is get increased, such type of this growth pattern, simply it is called as isodiametric growth. Such type of the growth pattern, these are called as the isodiametric growth. So you know very well, normally when we talk about any plant, so initially plants, these are the present like this. Now they plant again, they are showing, they become long, length will be increased like this. After few days, now it is become stem, it is become quite thicker. Leaf size also they get increased. And finally, now there is a formation of a large plant like this. So when we are talking about secondary growth means what? Secondary growth nothing but when plant is become mature. So whatever the tissue get added here, the lateral meristem, these are the side position exactly यहाँ पे. So initially, suppose these are the diameter of this plant. Now the diameter will be increased. Now it is become like this. अभी ऐसा बनेगा. Now again it will be get increased. Now it will become like this. So we are talking about increase in diameter. Slowly, slowly increase in diameter. Don't draw this diagram, it's for your understanding purpose. So if you see, during the initial developmental growth, primary meristem is very active, like root apical meristem, shoot apical meristem, intercalary meristem, they are very active. But when plant will become completely mature, sorry, Lisa D'Souza. Hmm. Yeah. So when we talk about Hmm. So when we are talking about the latest lateral stage of the plant, so just compare this one. Now diameter itna tha, now the diameter will be increased. Again diameter will be increased. So now what happened? Whatever the size will be increased, that size will be isodiametric growth. Means what? They are showing radial growth like this. Diameter increase ho raha hai na. So from all the side of the plant, that size will be increased or that diameter will be increased. That's why such condition is called as isodiametric growth pattern. Okay? Clear this point? What is meaning of isodiametric growth? Any doubt? During the latest stage of the plant, whatever the lateral meristems are present, they present on the side of the plant. They are responsible for they just increase the girth of the plant. So once plant is become mature, now slowly plant inside the plant wood formation get initiated. Now the cell wall it is become quite thick. Now toward the outer side bark also get formed. So now these are the some changes due to lateral meristem. मधुरा, नताशा, तन्मय, वरुण, मंगेश. Okay, चलो. So next part we are talking about exactly difference, and then we start with the one by one how that secondary growth is get initiated. Just try to imagine thing. Okay? Primary part is done. Now we assume one condition: plant is become totally mature. Now wood formation process get initiated. So we are talking about how that wood get formed, because again it is a part of the secondary growth. So first of all we are talking about in this chapter specifically we'll discuss secondary growth, and specifically secondary growth will study in case of the dicot stem as well as in monocot stem. Sorry, dicot stem and dicot root, because in case of the monocot it is totally absent. So in this chapter we are not taking gymnospermic stem and the root, only we are talking about dicot root and the dicot stem. Okay? Now, so when we are talking about such part, whatever we are talking about this part, now first of all we start with the information with respect to dicot stem. Dicot stem secondary growth. Chalo, write down the title. So initially we start with the dicot stem secondary growth. Please remember, secondary growth it is present in case of the gymnospermic plant and the dicot. But in this chapter, in our syllabus, we have to focus only dicot, root and stem. Gymnospermic plant it's not given, it is not part of our syllabus. But please mention one point: secondary growth also present in gymnosperm also. ठीक है, चलो, डन दिस पार्ट. Should I continue? So we are talking about next part, secondary growth, which is present in case of the dicot stem. First of all, we talk about the dicot stem and quickly we cover the dicot root also, because maximum question coming from the dicot stem. Chalo. Should I continue next part? Just quickly check the diagrammatic representation for the dicot stem. हमने जो last class में देखा था ना, previous classes we already discuss about the anatomy for the dicot stem. So just quickly check. Just open the previous pages and quickly check dicot stem diagram. And just read the some vascular bundle type. Dicot stem diagram and another one vascular bundle. Just read about the vascular bundle. Only in dicot. Dicot stem. ठीक है. Now first point. So before starting with the some steps, first of all we are talking again, we are repeating intrafascicular cambium and the interfascicular cambium. फिर से हम देख रहे हैं. So we are taking diagrammatic representation for TS, transverse section of dicot stem. And you know very well, inside the dicot vascular bundle which is present, that is present in a ring form. So ring form vascular bundle, V bundle, I'm writing just V bundle. So ring form vascular bundle is present. Am I right? Correct है. Ring shape में था ना vascular bundle in case of dicot stem? Yes or no? Ring shape means what? They arrange in a circular manner. Huh. Okay. And in case of the monocot vascular bundle, they scattered all over the ground tissue, small size, large size. But in case of the dicot, ring formed vascular bundle is present. So quickly we try to represent those structure. Suppose ये outermost layer है, outermost layer. Inside the outermost layer, we are talking about first part, that is intrafascicular. So you know very well, one xylem, phloem, vascular bundle है. Now these are the another vascular bundle. Now these are the next one. So there is a presence of the few vascular bundle. Now that vascular bundle, now they get differentiated with the help of this red color. Okay? So could you tell me that red color, this red color, nothing but these are called as what? Red color क्या है? Red color, this is one cambium. What is the specific name for that cambium?
Intrafascicular.
Yeah, intrafascicular cambium. Intrafascicular cambium. Why we are talking about the intra? Because they are present between the xylem and the phloem. So for your simplicity, I just remove one vascular bundle. Inside this vascular bundle, एक red color की line है, that is called as the intrafascicular bundle, which is a part of the cambium. Toward the outer side, here there is the presence of the primary phloem, P for the phloem, and now there is a presence of the primary xylem, X for the xylem. So quickly we are representing this one part of primary phloem and this one part of the primary xylem. Just represent only one. If you want, you can draw outside also. कोई problem नहीं है. So toward the inner side, now there is a presence of the primary xylem, primary X, and toward the outer side, now there is a presence of the primary phloem. And we are talking about the dicots, so obviously cambium present. And which type of the cambium is present? Intrafascicular cambium, because inside this single vascular bundle, single ring, single vascular bundle, xylem and phloem, they differentiated with the help of the cambium. That's what is called as the intra. Got it? तो ये हो गया इंट्रा fascicular cambium. Now, next part we are talking about interfascicular cambium. So now what happened between these two cells like this? यहाँ पे कुछ cells present होंगे in this point. So here we are talking, we are giving this some dotted line. Now there is a presence of some specific type of the cells. This one, these are called as medullary ray. These are the specific type of the tissue. These are called as medullary ray. Medullary. Now, what is the specificity with respect to this medullary ray? Medullary ray, basically, they are parenchymus tissue. They are parenchymus tissue. I'm writing just parenchyma. Now, what happened? Such medullary ray, during the secondary growth, now they converted into secondary meristem. They converted into secondary meristem. So now, could you please tell me? Medullary ray, these are the parenchyma. Parenchyma means what? Medullary ray, basically, these are the primary permanent tissue. Primary permanent tissue, they undergo the process of dot and they form a secondary meristem. Can you tell me which process? Differentiation, de-differentiation, or redifferentiation? Anyone? Yes, Sakshi. So medullary ray, basically those are the example for the parenchyma tissue. Parenchyma means what? ये permanent tissue है, because medullary ray, they are parenchyma, they are responsible for the storage of the food. So they are having specific kind of the function. So that is a permanent tissue. But now what happened during the secondary growth? Such medullary rays, those are the primary permanent tissue. Primary permanent tissue, they undergo the process of de-differentiation. Means what? They regain division capacity. They undergo the process of de-differentiation and finally they form secondary meristem. And please remember, whatever the secondary meristem they get formed, that secondary meristem, simply it is called as interfascicular cambium. It's called as inter. Why inter? Because it is present between two vascular bundle. Interfascicular cambium. And why we are talking about the inter? Because they present between two vascular bundle. ठीक है? They present between two vascular bundle. So please, important point क्या है यहाँ पे? Interfascicular cambium as well as the interfascicular cambium. Inter and the intra. So initially medullary rays are present. That medullary ray, they undergo the process of the de-differentiation. After the de-differentiation process, they convert it into secondary meristem, and such secondary meristem, now they are called as interfascicular cambium. Got it? Clear this point? Any doubt? Any doubt? साक्षी, अरे बोला ना लवकर, तुम्ही तुम्ही काही बोलतच नाही बाबा. वरुण, वियांका, यश, क्या बात है? घर पे कॉल आया था क्या? आज बराबर 50 है लोग. हाँ, चैत्रा, did you get any call from office? Aditya, ठीक है. So remember intra and the inter. ठीक है. Now we are taking step by step information how that vascular bundle further they get developed, because just clearly we have to take one point, how that wood get formed, how the outer side bark get formed. So we are talking about that part because that is all related to secondary growth. So one by one we are talking about the few steps, and during the few steps, first of all we are talking about formation of vascular cambium. First point. So I hope you remember when you start this chapter, under the lateral meristem हमने कुछ example लिखवा के दिए थे. Under the primary origin, one example intravascular, secondary origin, vascular cambium, bone cambium, intervascular cambium, ring cambium. So now we have to focus on the vascular cambium, which is a part of the dicot stem. Now what happened? Vascular cambium, they
Excuse me,
get
ma'am.
formed. Yeah,
Ma'am, could you please explain the previous part again?
which one?
Ma'am, the
Inter and intra.
interfascicular cambium. Yes, ma'am.
Because when I started this point now, so many students missed this class due to our exam or whatever. What is the reason? Chalo. So listen, these are the, take a one stem, mature stem, take a one thin slice, because we have to observe transverse section under the microscope. That thin circular slice we keep under the microscope, and we try to understand how the vascular bundle they get formed, means how they are showing the further development inside that vascular bundle. Now we are talking about this blue color. Blue color is a single vascular bundle who consists of the xylem as well as the phloem. Xylem as well as the phloem. Okay, this part, these are the called as the vascular bundle. So I am just writing VB. Okay, chalo. So these are the vascular bundle. We are talking about the dicot. Obviously, cambium present between xylem and the phloem. So that's why red color line, these are the cambium. Above this red color line, there is a presence of the primary phloem. Below the side, there is a presence of the primary xylem. Because now between the primary phloem and the primary xylem, cambium is present, and that cambium specifically it is called as the intrafascicular cambium. Why intra? Because these are the part of single vascular bundle. Cambium present inside the single vascular bundle. The differentiated primary phloem and the primary xylem. So primary in origin, intrafascicular. Now what happened? Outside this, we draw some line, this dotted line. So between two vascular bundle, there is a presence of the medullary rays. Medullary rays kya hai? These are the one parenchymal tissue responsible for the storage of the food. Means these are the primary permanent tissue. Where these are the present? Between two vascular bundle. Blue color का जो draw किया है उसके outside. So between two vascular bundle, medullary rays are present. Medullary rays, they regain division capacity, that is called as the de-differentiation. We already discussed this part during the initial class. De-differentiation means what? Regain the division capacity, and due to de-differentiation, primary permanent tissue, that is medullary ray, they converted into secondary meristem. And you know very well, meristem means what? Active cells, metabolically active. They are produce all type of the cells which is present in plant. So it is become meristem. It is behave like a stem cell. And such kind of this secondary meristem, simply it is called as interfascicular cambium. What is meaning of the inter? Because they present between two vascular bundle. Between two. That's why we are using word inter. Got it? So
Yes,
these are
ma'am.
the two important parameters. Number one, intrafascicular cambium. Another one, interfascicular cambium. Done?
Yes, ma'am.
Yeah.
Thank you, ma'am.
So next part, we are talking about formation of the vascular cambium. Very, very, very important. Now, remember this point. When we are talking about the vascular cambium, basically these are the partly primary in origin and partly secondary in origin. So we are using specific word partly, means partially. So what is meaning of that? Primary in origin and the secondary in origin. So simple, I'm giving you some equation. Intrafascicular cambium intrafascicular cambium plus interfascicular cambium. Inter plus intra. Both this cambium, they join with each other. They join with each other, and after the joining, they form a typical structure. That structure basically is called as the vascular cambium. What they form? They, after the joining, they form a vascular cambium. So intra and interfascicular cambium, they join with each other, because you know very well, when we already discuss about the intrafascicular cambium, these are the primary in origin. Just check the initial classes. Interfascicular cambium, these are the secondary in origin, right? So now what happened? That's why we are using specific word, partly primary and partly secondary in origin, because now intra and the interfascicular cambium, they join with each other, and now they form a ring. That ring simply it is called as vascular cambium. Hmm? So quickly we are taking the diagrammatic representation. Same diagram we are taking. At the center, now there is a presence of the intra. Now toward the outer side, there is a presence of the inter. And finally, what happened? That inter and intra, they join with each other like this. And after the joining, they form a specific type of the cambium that is called as vascular cambium. So just give a dark line with pencil. So these are called as the vascular cambium, which is a present in a ring form. Ring shape, basically ring form. That's it. Chalo. So these are the first point, formation of the vascular cambium. Done. Sakshi, Yash, Amal, Madhura, Arya, Rishikesh, Sam. Done ना. ठीक है चला. So नंतर second point, activity. चलो. So ये हो गया first point with respect to formation. Now we are talking about number second point, activity of vascular cambium. To the number two, first point, formation of the vascular cambium, done. Now we are talking about activity of vascular cambium.
Ma'am, please scroll up.
Diagram. Hmm. Sanika. Hmm. Done.
Yes, ma'am.
Chalo. Activity of vascular cambium. So just stop writing and listen carefully, because ये step बहुत important है, because you have to remember sequence from outer to inner, and यहाँ से बहुत सारे questions design होते हैं. So please stop writing. I'll give the time. So activity of vascular cambium. Vascular cambium, it is a part of the secondary meristem. Secondary meristem means what? They can form a new cells of the xylem, new cells of the phloem. So now what happened? At the center, suppose we are taking a very small section of the vascular cambium. So these are the vascular cambium. It is present in ring form, but I'm taking very small section. So these are the presence of the vascular cambium. Now what happened? Such vascular cambium, some cells, not all, okay? So we are talking about some cells, they cut toward the outer side. Some cell, they cut toward the outer side, because cut means what? Now they get differentiated, means redifferentiated. So vascular cambium, some cells they cut, and now toward the outer side they form secondary phloem. Okay. Similarly, some cells of the vascular cambium, they cut toward the inner side. Not all, some cells. And now toward the inner side, they form the secondary xylem. What they form? The secondary xylem. But please remember, now what happened? Vascular cambium, it is the one kind of the secondary meristem. They can form a new xylem, they can form a new phloem, but now they are secondary xylem and the secondary phloem, because vascular cambium, it is a part of secondary growth. Vascular cambium, part of secondary growth. That's why they cut toward the outer side, so they can form a secondary phloem. They cut toward the inner side, now they form a secondary xylem. But now what happened? If we compare this point, out of secondary phloem and the secondary xylem, which cells formation rate is more? So please remember, neat question. Secondary xylem formation rate, it approximately 8 to 10 times more. Formation 8 to 10 times more active than secondary phloem. Question: how many times? 8 to 10 times secondary xylem they get formed. For example, if one cells of the phloem get formed, means secondary phloem, that time 10 cells, approximately 10 cells of secondary xylem get formed. And why such kind of this variation? Because secondary xylem, they provide mechanical support during the aging. Because you know very well, plant it is become vascular. Now they are having high molecular weight, so they required extra support. And who provide the support? Secondary xylem, because secondary xylem also called as the wood. If you remember, हमने वहां पे देखा था, xylem also called as the wood, right? Fiber भी present होगा. Whatever the cells are present, which is a rich with this sclerenchyma tissue, dead tissue, all they are involved during the provide the mechanical support. So we are talking about the reason why such kind of this condition is present, because secondary xylem simply they provide mechanical support during the aging time. That's why, as compared to secondary phloem, secondary xylem rate is more faster. Please remember this point, and if you want, you can write down one point. Secondary xylem, nothing but, these are the indication, these are the signal that inside that plant wood formation it is get initiated. And once wood is get formed, it means that that plant is become mature plant, they are showing secondary growth. Got it? So secondary xylem, nothing but we are talking about inside the cells wood get formed. Chalo, you can complete this part. In third part, we'll continue. We'll see the diagram. Exactly what is the position of the primary phloem, primary xylem, secondary phloem, and the secondary xylem, along with vascular cambium. We'll continue with third part. Should I continue? Tell me, okay, once time. Should I continue with diagram? Third point. Second point, activity of vascular cambium, and these are the information. Clearly, we mentioned one point. Once vascular cambium get formed, now slowly they get redifferentiate. Obviously, some cells cut and they form secondary phloem, means they get redifferentiate, and now they can form secondary phloem and the secondary xylem. Lisa, done? Very go. Aditi. Okay. So next part we are talking about number third. What is the exactly arrangement of different cells with respect to cells of vascular cambium? Cells of vascular cambium. So in this part we discuss about how they get arranged. So I'm not taking too much complicated part. I'm just simply telling you vascular cambium they can produce. क्या क्या produce करेंगे हम उसके बारे में बात करें. Okay, so that's why we are talking about cells of vascular cambium. They can produce secondary xylem, as well as they can produce secondary phloem, and along with that they can produce the another important cells, that is secondary medullary ray. Secondary medullary ray. Okay, you know very well the function of the xylem, basically water conduction, function of the phloem, food conduction. But now secondary medullary rays, whatever these are the present, such kind of the cells, basically they are responsible for radial conduction of water, food and the gaseous. They're responsible for the radial conduction of gases, food as well as water. And the another point, when we are talking about the secondary medullary ray, they present in a narrow, basically these are the narrow band. We'll see in this part in diagram. So medullary ray, basically nothing but these are the narrow band. Present between secondary xylem and secondary phloem. So these are the something a narrow band, that is only we are talking about medullary ray. So cells of the vascular cambium, basically majorly they can produce the secondary xylem, secondary phloem, as well as they produce the secondary medullary ray also. And the major function, they are responsible for Radial conduction. We already discussed what is meaning of the radial. Radial means what? Horizontal conduction. Whatever the surrounding cells are present, all the surrounding cell they get water, food as well as the gas from secondary medullary cells. Okay, and these are the some narrow band which is present between secondary xylem and the secondary phloem. So in diagrammatic representation, we'll discuss this part. Then you will get the clear cut idea. Chalo. Should I continue with diagram? Initially use pencil, please. So we are talking about at the center, this one, there is a presence of the vascular cambium. Quite thick because it's cambium. Hmm. Vascular cambium. Okay. Now I hope you remember vascular cambium. They cut toward the outer side as well as the inner side, and they form secondary xylem as well as the secondary phloem. So quickly we are talking about toward the outer side. This one. The cells are present. These are called as secondary phloem. I'm giving just cross, okay? If you want, you can use star dot, your choice. So exactly toward the above side, some cells of the vascular cambium they cut and they form the tissue that is secondary phloem. And you know very well, primary phloem it is already present, right? So quickly we are talking about toward the outer side. Now there is a presence of the another cell. Cross ही देते हैं चलो better. So now there is a presence of the another cell that is primary phloem. Because primary phloem it is already present. But now what happened? Due to activity of the vascular cambium, now there is a formation of the secondary phloem. So this part, these are called as the primary phloem, which is already present, right? Don't change the sequence. I'm giving the number. ये number 1 है. These are the number 2. These are the number 3. Because now we are taking outer to inner side. Question on the sequence. Vascular cambium start from the vascular cambium. Vascular cambium they cut toward the outer side. They convert into secondary phloem. Already primary phloem is present, which is a part of the primary growth. तो हमें उसको भी draw करना पड़ेगा. So again toward the outer side there is a presence of the primary phloem. Now same vascular cambium they cut toward the inner side and they form secondary xylem like this. And you know very well the rate of the secondary xylem formation is quite higher, 8 to 10 times more as compared to secondary phloem. So exactly below there is a presence of the secondary xylem, which get formed. So you can draw the compact more cell because activity formation rate is quite higher. So if you want you can draw 1 to 2 layer, no problem. Your choice, okay? अगर ज़्यादा diagram complicated हो रहा होगा so just you can use some highlighter or some sketch pen or something pencil color and you can represent like that, no problem. So this white color, nothing but all these are the secondary xylem. And you know very well, already primary xylem is present, right? So this green color, nothing but these are the part of the primary xylem. And you know very well, at the center of the cell, at the center of the stem, now there is a presence of pith, P-I-T-H, which is present at the center. So just check the sequence. Number 1, primary phloem. Number 2, secondary phloem. Number 3, vascular cambium. Number 4, secondary xylem. Number 5, primary xylem. And finally, on the number 6, there is a presence of the pith. So these are the exactly arrangement from outer to inner. Because in NCERT it's quite complicated diagram, but from this chapter actually diagrams से question नहीं बनते. So whatever the questions are coming, those are related to concept. So you have to focus on concept.
Ma'am, please zoom out.
ठीक है.
Done, ma'am.
Any doubt from the vascular cells? Yeah, quickly tell me any doubt. This chapter is required quite practice. थोड़ा सा मेहनत चाहिए because I know थोड़े से terms अलग अलग हैं. You know the basic cells like parenchyma, collenchyma, sclerenchyma. So these are the something different because obviously it's a part of NEET, part of the 11th standard. तो syllabus थोड़ा सा difficult होगा. Once you understand it's very easy, but I'll tell you for the secondary growth you require more practice. इसको बार बार learn करो. If you're not understanding, then try to solve the NCERT. You can ask me, you can solve the questions also, you can listen again recording classes also. Your strategy. Janil, should I continue next? Now I just want to represent a narrow band. Okay, chalo. Because just we mentioned here, vascular cambium, they produce secondary xylem phloem, as well as along with that they can produce a secondary medullary rays also. Those are present in a narrow band. So quickly will represent exactly where such narrow bands are present. कहाँ पे present होते हैं? They are present between primary xylem and the primary phloem. So just check, use a pencil, use a highlighter, and just give a band shape. That's it. So these are the blue color and the white color. So we are talking about medullary rays. They are present here. Like this. Narrow bands. Parenchymus in nature, obviously, because it's a medullary ray. And as well as they can cross secondary xylem and the secondary phloem. So this green color, these are nothing but we are talking about secondary medullary rays. Those are present in a narrow band and responsible for conduction of gases, conduction of food as well as water, but in radial manner, in horizontal manner. Chalo, done. Should I continue next part? Arya? Hmm. Chalo. So 1, 2, 3. Now we are talking about fourth step. What is the exactly fate of primary xylem and primary phloem? Fate of. It's very important. Just listen carefully. Now, what happened? Due to activity of the vascular cambium toward the outer side and the inner side, some cells they get cut and they convert it into secondary xylem and the secondary phloem. If you see the exactly position near the secondary phloem, now primary phloem is present, and you know very well primary phloem means what? These are the part of the primary growth. They are delicate as compared to secondary phloem, or what we say these are the less active. Similarly, secondary xylem they are more active because 8 to 10 times more times they get divided. So now what happened? Slowly, primary phloem and the primary xylem they get crushed due to activity of secondary and the secondary xylem. Due to activity of secondary xylem and the secondary phloem. So जैसे जैसे expansion होगा toward the outer and the inner side, so primary phloem they get crushed, even primary xylem they get crushed. So we are talking about fate with respect to primary phloem and the primary xylem. So first of all we are talking about number one, what is the exactly fate with respect to primary xylem? What happened with respect to primary xylem? One question. In case of the what we say, that is in case of the primary xylem part, basically you know very well primary xylem it is a quite hard tissue. These are the quite strong tissue, right? And why? Because inside the xylem tracheids are present, vessels are present, fibers are present. All these three there, these are the dead tissue. Dead tissue means what? Sclerenchyma tissue. Obviously they quite hard. But now what happens? Sometime primary xylem they push toward the inner side because secondary xylem cells they are more active. 8 to 10 times they are more active. So slowly what happened? They start pushing primary xylem towards the center. और center में कौन present है? Tell me, at the center which cells are present? Pith cells are present. Those are the living, those are the delicated, those are the parenchymatous tissue. So that's why now finally pith start crushing. So just take this step by step what is happening. So first of all we are talking about what is the exactly fate with respect to primary tissue. Primary tissue basically these are the quite strong tissue because primary xylem out of 4 cells 3 cells these are the dead. Another one. Now what happened? We are talking about next step. Due to activity of second due to activity of secondary xylem, now slowly primary xylem they push towards the center. Slowly they push towards the center. Just check the position. Vascular cambium exactly below secondary xylems are present. Secondary xylem 8 to 10 times they are more active. So they are showing rapid division. तो वो क्या करेंगे? Slowly they push primary xylem toward the center. And now what happened? Due to that pushing phenomena, at the center teeth are present. So due to such pushing process, now it will be start crushing of pith cells. Crushing of pith. And why? Because you know very well pith basically these are the parenchymous in nature. They having the thin cell wall. major function storage of the food. So as compared to xylem, they are quite delicate, they quite soft, so obviously they will get crushed. And now what happened at the end, slowly whatever we are talking about, the interior region of the stain, interior region of stain, now they filled with, secondary xylem. That is, we can say now these are the stage where wood get formed, because secondary xylem also called as the wood. So just check the steps, what is happening, what is the fate with respect to primary xylem. First one, xylem, primary xylem, whenever we are talking about, these are the strong tissue. Why strong? Because tracheids, vessels and the fiber, these are the three dead tissue, those are made up of the sclerenchyma. So obviously they are not easily crushed. But now what happened, due to activity of the secondary flow xylem, due to activity of the vascular cambium toward the inner side, formation of the secondary xylem. Secondary xylem, they form 8 to 10 times faster. Ultimately what happened, they start pushing the primary xylem, because हमने sequence में देखा था, यहाँ पे present है vascular cambium, exactly below the vascular cambium. Now there is a presence of the secondary xylem, and now there is a presence of the primary xylem, and at the center यहाँ पे present होगा pith. So now what happened, secondary xylem इसको push कर रहा है. Now together they can start pushing the pith, and slowly whatever the pith present at the center, they start crushing. Crushing हो रहा है मतलब क्या हो रहा है, they become dead, they undergo the senescence process, right? So slowly due to that pushing process, the entire region, means in central region, interior part of the stain, it totally filled with the secondary xylem. Remember this point, बाद में primary xylem also they get degrade, because secondary xylem they push. So initially pith also get degrade, and as well as the primary xylem also they get degrade. Simple phenomena. And center of that part, center of that stem, they completely filled with the secondary xylem. That time you can say yes, now inside that plant wood get formed. Got it? Wood formation. Clear this point? Any doubt from fate of primary xylem? Quickly tell me. Sorry. Should I continue with next part? Primary phloem. Heram Prabhu Desai. Sanika. Padma.
Yes, ma'am.
Hmm. Okay, chalo. Next and the simple step and last point. Just 2 minutes, okay? So we are talking about what is the exactly next part, that is fate of secondary, sorry, primary phloem. इसी continue कर रहे हैं. So fate of primary phloem. Obviously, phloem part है, which is a reach made up by the Seuss tube cells, then the companion cells, now the parenchyma, and the fiber is present. So it's quite weak, as well as these are the thin tissue. So result क्या होगा? Obviously, due to activity of secondary phloem, means pushing everything. So due to activity of secondary phloem, now slowly whatever the primary phloem tissues are there, they get crushed. That's it. So no question from the primary phloem phase. Whatever the question coming, that is question coming from the wood formation, because during the wood formation, now internal, whatever the internal part of the stem is present, because it's mostly rich with the pericycle, pith, etc. So all those cells they get crushed, and interior it is totally filled with the secondary xylem. And once it is filled with the secondary xylem, now clearly we can say yes, inside that plant wood formation process is get initiated, that plant become mature. Yeah, that's it. So any doubt from today's class? Any doubt?